All right, hello IB Bio students. This video is about neuron function. So whenever we're talking about functions in biology, a central theme is that we need to understand the structures first. So neurons are a kind of cell. They're a really highly specialized cell. In this diagram, you can see there are two neurons. Like all cells, they have the organelles that they need to carry out their functions. So they have a nucleus, they have endoplasmic reticulum, they have mitochondria, and all of those different organelles. Now, things that's unique about a neuron is the organelles are mostly centralized in the region called the cell body. And then they have these extensions coming out, which are called fibers. The shorter fibers are called dendrites, and the long fibers, usually there's one really long fiber, is called an axon. And here you can see there's a zoomed in box there in the bottom left showing a synapse. A synapse is just the junction between two neurons. So today we'll be talking about how electrical impulses move along fibers in neurons, mostly thinking about axons, and we'll also look at how a synapse works, so how an electrical impulse reaching the end of one neuron can trigger an electrical impulse in the next neuron. So when I say electrical impulse, what I'm really talking about is an action potential being propagated along a neuron like a wave. And so what an electrical impulse is actually an action potential happening sequentially along, let's talk about the axon, along an axon. So an action potential will happen at one location, and that will trigger an action potential to happen at the next location, and the next location, and so on until it reaches the end of the axon. An action potential is usually understood in terms of the change in membrane potential or voltage across the membrane. Voltage is referring to a difference in charge. And so in this graph, you can see how the charge or potential changes, or the voltage changes across a neuron membrane when an action potential occurs at a specific location. And so there's a pattern of change from what's called the resting potential to a gradual rise up to a threshold potential, which then triggers the depolarization and repolarization of the action potential. There's a short undershoot and then a return back to resting potential. So our goal here is to understand what's happening at each of those phases represented in this graph. This diagram is showing the plasma membrane of, let's say, a region of the neuron. So maybe it's actually showing a region of a dendrite, but you can imagine the same thing existing in a region of the axon. And you can see there's different kinds of proteins in the plasma membrane. So the orange region is the inside of the neuron, and the blue region is outside the neuron. And in between we have the plasma membrane with various proteins in there. The purple protein is the sodium-potassium pump. And the orange one is a always open potassium channel. And the sort of yellowish one is an always open sodium channel. Those aren't the only kinds of proteins in plasma membranes, but those are the ones that are significant for creating and maintaining the resting potential. So when a neuron is resting, it has what we call a resting potential, which is roughly minus 70 millivolts. And when we say that, we mean the inside relative to the outside is negative, the inside of the neuron. The main explanation for how the resting potential is created and maintained is through the sodium-potassium pump. So the sodium-potassium pump uses ATP to actively transport 3 sodium ions out for every 2 potassium ions it pumps in. So since it's pumping out more positive than it's pumping in, the outside is more positive relative to the inside. You can also say the inside is more negative relative to the outside. It basically means the same thing. So that's the main contributor to the resting potential, is the sodium-potassium pump. But there are other contributors which you should know about as well. So a second contributor are these always open potassium and sodium channels. And so they sort of undo what the sodium-potassium pump does. Now, if there were lots of both, it would be meaningless. It would really undo all the work of the sodium-potassium pump. But there aren't so many of them, and there's a lot more potassium channels than sodium channels, and that difference is important. Since we have a lot more potassium channels, the potassium that we pumped in is allowed to escape. We have a few sodium channels, so some sodium is allowed to come in, some of the sodium we pumped out, but not that much compared to the potassium. So we're pumping out more positive than we're pumping in, right? So we're pumping out 3 sodiums for every 2 potassiums in. But then a lot of the potassium that we let in, not all of it, but a lot of it, is able to diffuse out again through these potassium channels, way more than sodium that comes back in. So that further amplifies this resting potential. Another factor to be aware of that contributes to the resting potential are other ions and other charged particles. So we have, for example, chloride ions within the cell. They are negatively charged. And we also have negatively charged proteins that exist within the cell as well, which further contribute to this negative 70 millivolts resting potential. Okay, so we create the resting potential. We maintain that because that is the potential energy that is used to make the action potential possible. So starting at the resting potential, we're now focusing in on voltage-gated ion channels. That doesn't mean the other proteins don't exist. They're still there. There's still sodium-potassium pumps and always open sodium channels and always open potassium channels. But now we're focusing on the voltage-gated channels because those are what cause the depolarization and repolarization of the action potential. So initially, we have to get this rise in potential to a threshold to cause voltage-gated ion channels to open. I'll get back to that number 2, why that rises later, but I'll just say that's due to what we call local currents. So as a result of local currents, the charge begins to rise. It reaches a threshold. That triggers the voltage-gated sodium channels to open first. So that's represented by the sort of yellow protein in this diagram. When they open, sodium rushes in because remember it was pumped out during the resting potential by the sodium-potassium pump. So sodium rushes in. So the inside of the cell becomes more positive relative to the outside. That's called depolarization. Here it's called the rising phase of the action potential. Same thing. After we reach a certain voltage, it triggers the voltage-gated potassium channels to open. That causes potassium ions to then rush out. Remember, they were pumped in. And yes, some of them escaped, but there's still more inside than outside. And so they rush out, and therefore the inside becomes more negative again. We call that repolarization. In this diagram, it's called the falling phase of the action potential. That will usually continue to decrease the voltage to below resting potential. We call that an undershoot. And then the sodium-potassium pump returns the potential, the voltage across the membrane back to the resting potential. Now, remember I said for that second point in the graph, that rise to the initial threshold before the depolarization of the action potential, I said that was caused by local currents. So let's talk briefly about what local currents are. Basically, you can see here in this sequence, this is looking at a section of the axon, you can see that action potentials at one location trigger the action potential in the next adjacent location. And the reason that is when sodium ions rush in and potassium ions rush out during an action potential at one location, they don't just stay where they are, they will move laterally or along the axons, both inside and outside, from an area of high concentration to low concentration. So they move by diffusion, and as they move over, they cause this slow, it's not really slow, but slower than the action potential, this slow rise in membrane potential in the adjacent regions next to where the action potential has just occurred. So we call that local currents, the diffusion of sodium and potassium ions both inside and outside the cell that follow an action potential at a given location, and those cause in adjacent locations this rise in potential to the threshold. And that will continue along, causing action potentials to occur in sequence. Now, the reason they only occur in one direction and don't go forwards and backwards at the same time is because when an action potential occurs, there is a short period after it occurs where the voltage-gated sodium and potassium channels do not open. They remain closed, and that's called the refractory period. And because of that refractory period, action potentials can only move in one direction. Another thing to note here is neurons are sometimes myelinated. In humans, some neurons are myelinated, some are not. It means they have these insulated portions covering them. They're called Schwann cells. Schwann cells are highly specialized cells that are mostly made of this protein called myelin. And so we call this whole area, these Schwann cells all together that cover an axon, we call that a myelin sheath. And they prevent action potentials from happening where the Schwann cells are. Now, in between the Schwann cells, there's a region that is not insulated, not covered by myelin. That's called, those regions are called the nodes of Ranvier. And basically the action potentials in a myelinated neuron only happen at the nodes of Ranvier. And so we get what's called this form of conduction called saltatory conduction, where the action potentials jump from one node of Ranvier to the next. And that is a faster way of conduction of action potentials because they happen at fewer locations. They're not happening one after the other. They're jumping a further distance each time, which is more efficient. All right, so action potential gets to the end of the neuron. Then the last thing left to explain here is, well, what happens when an action potential gets to the end? How does it trigger or influence the neighboring neurons? So this is called synaptic transmission, and we're looking at the junction between two neurons, which we call a synapse. And you can see in the diagram, the top bit is the end of the axon of the neuron where the action potential is coming in. We call that the presynaptic neuron, and the neuron that is being affected by that we call the postsynaptic neuron. So basically what happens, action potential reaches the end of a neuron, the presynaptic neuron. That causes voltage-gated calcium ion channels to open. So there's a different kind of voltage-gated channel in this region of the neuron. And calcium ions, which were pumped out previously, rush in by facilitated diffusion. That causes these little vesicles that contain neurotransmitters called synaptic vesicles to migrate to the membrane of the presynaptic neuron, fuse with that membrane, and exocytosis occurs, causing the neurotransmitters to be released into the space between neurons called the synaptic cleft. Then the neurotransmitter will move by diffusion across the synaptic cleft to the postsynaptic membrane and bind to ligand-gated ion channels in that membrane. Depending on the neurotransmitter, it could have an excitatory or inhibitory effect. If it's an excitatory neurotransmitter like acetylcholine, for example, it'll bind to the ligand-gated ion channels, causing them to open. And in the case of acetylcholine, the ligand-gated ion channels allow sodium to come in. And so as sodium rushes into the postsynaptic neuron, it causes a rise in charge, which then, if it reaches the threshold, you can imagine that that would trigger an action potential in the postsynaptic neuron. That would be the effect of an excitatory neurotransmitter. There are also inhibitory neurotransmitters, which would have the opposite effect. After the neurotransmitter does its job, an enzyme is released into the synaptic cleft, which breaks down the neurotransmitter. and then the products of breakdown are pumped back into the presynaptic neuron, where they are reassembled back into neurotransmitters, put back into synaptic vesicles again, allowing the process to happen again. We're basically resetting the presynaptic neuron, resetting the synapse. The other thing that has to happen to reset the synapse is calcium ions need to be pumped out. So they're pumped out, and then the synapse is ready for this synaptic transmission to occur again. I think I got everything for synaptic transmission. Let me just look this over. Yeah, that looks good. So this is the last slide. That's not bad, 15 minutes. I hope this is helpful for you to understand neuron function. Thank you.